Thank you, Jason. Thanks a lot for inviting me and for the nice introduction. So I'm going to give you a bit of an insight to image analysis, but before we start, what do you think, what is image analysis? Just let's make it a bit interactive. Let's make the question a little bit different. What is the input of an image analysis algorithm?
Well, probably the data you got from your camera or photo camera.
I wouldn't mind if you raise your voice a little bit.
Okay. So it's probably the data you get from your CCD camera or photo multiplier, photo counter.
What would be the output? Is it an image? It's maybe numbers. Let's put it like that. Okay. So, and I would like to show you what's in between getting an image and getting something wrong. But before going there, let me show you why I believe computers are important. So just will show you three standard my images, which maybe highlights why observing images is important and why using computers are also important. So what do you see on this image? Those who know say. It's damn difficult, right? Why? Because we have no idea what we are looking for. Do you see the animals on that picture? No. Do you see this horse there? Come on. Okay, so this is a very different. There are horses there. So there is one head there, another one there, maybe another one over there. And the ground becomes very, very much more simple if I ask you, do you see the Dalmatian shadow on that picture? Immediately, right? Because we have prior information about the shape that we are looking for. And let me show you another picture. What's the question here? So the question usually is which rectangle in the middle is bigger? Maybe you know it from the textbooks or triangle. And what's the answer? You sure about that? No, right? Because we don't really count measurement in our eyes, so we can't really be sure. They are actually the same. Okay. And one more picture. So human eye and the human brain is especially trained for recognizing faces. Actually, a very big part of it is for that. Can you recognize the faces there? A lot, right? How many? Don't even count it. It's 14 or 15 or something. So imagine a computer program which could count it. It's an absolutely difficult thing. So what to take home from here is there are questions which are very difficult, even for us. If we know something prior, it becomes easier. Or eye is not really tuned for quantitative measurements, but there are things that we are very good at. Okay? And I would like to show you how bad we are in certain other things. So these are cells stained with some further staining the DNA. How many cells do you see here? About 10, right? It takes, for a human, it takes quite some time. For a computer, it's much bigger. How about here? Very good. Okay. And the good thing with these very high-throughput computers is that I can go further and even further. And using these really high-throughput machines, imagine that we can acquire thousands of cells. And the scenarios we do nowadays, we easily go to 10 million to 10 billion to 100 billion cells, analyzing each of them separately. So is a need for objective quantitative analysis, and the amount of data that we have is really large. So we need to handle it somehow, which means we need to do our automated analysis. So what I'm going to talk about, a few basic definitions: what is image processing, what is histogram transformation, multiple morphology, filtering, segmentation, shape and feature extraction, tracking and convolution. These are standard themes. I'm not going to much deep into it, just to give you an overview. And what you should see is, if we have a computer, a microscope, we take an image, and if we want to quantify it in numbers, and this way how to get from here to there involves all of them or some of these steps. So, and the first thing what we are going to see is how to manipulate the image itself. Next one is how to get objects from the image. And once we get the objects, what to measure them. And if we measure them, then we have numbers. Okay, so what is an image? So if I say resolution, what's the typical resolution, for example, of your pocket camera? Like, I don't know, like this camera. So this smartphone camera. This is megapixel. What is a megapixel? What is a million pixel? Which means it's approximately, it's exactly 1,000 by 1,000 square, right? So we have like 1 million measurement. Maybe your screen is about that size. Then, very important thing, bit depth. So bit depth is a measurement. Well, it's actually the way how we represent data. And we represent it on bits. A bit is either there is a signal or there is no signal. If there are a collection of them, we can store more information. If there are a lot of them, we can store even more information. These cameras, what we usually use in microscopes, are in a range of 12 to 16 bits, which means we can distinguish between usually 4,000 to 16 million different levels of information. Spectral channels, I think you heard about that. In image analysis, there is a single and there are multiple, like RGB, or in microscopy we have several channels. Maybe you heard about satellite imaging, there are hundreds of channels there. And finally, which is very important, I believe, is compression and formats. So what comes immediately to your mind if I say compressed image? JPEG, right? So JPEG uses a very interesting phenomenon, is that human senses colors in a different, well, senses information in different color channels differently, and there are more or less important colors, and based on that we can compress certain colors more than others. And in this way we can compress the data to a factor of 10 easily. Okay, and there are also lossless compression. Lossless compression means if I compress my data and decompress, I don't lose information, whereas in lossy compression, I cannot recover back my original data. Okay, next thing is a digital image. So digital image is composed, as already Gabor said, an Excel table. We can consider a digital image as an Excel table where every single information bit is stored as several numbers or one number. If it's several numbers of computers, then we can represent colors. If it's one number, we represent intensities. The general idea is that smaller colors represent darker bits and higher numbers are brighter. So given that, what do you think? What's this image? It's you can see as an Excel table. What would be that for a computer? Or for you, if you don't do that visualizing. So these are darker corners here, right? These are brighter ones. If I show you what we would see. Okay. So that's the idea of how a computer represents. And now, if we know how it's represented, maybe we can start to work on it. So to create an image, there are two very important phenomena. One is sampling. So sampling There is an important thing. I don't know. Did you already talk about the Nyquist theory yesterday? Or today? No? Okay. So there is a very important theory called the Nyquist-Shannon sampling theory. In principle, what it says, if you want to represent changes in the image not bigger than a given size, then you need to do sampling twice as much as your smallest sample that you want to represent. And to show you in practice what it means, let's say we have sampling points with these dots, and we have signal what we want to sample. So imagine if the signal was very dense, but we only have like 20 points, we start to introduce artifacts. The same for images. If you have, for example, a firewall, and you have a proper sampling, you see the breeze. If you don't do that properly, then you start to see the artifacts. It's very much the same for biology or microscopy. If you have patterns in your sample which are too dense, you need to have proper sampling. The next thing, what is very important, now we understand how to create the dots in the Excel sheet. Now what we need to know is how to put the numbers into that Excel sheet. And this thing is called quantization. So quantization is a process which approximates values from the continuum into the digital world, and very intuitively we can make a linear correlation between the observed light and the represented number. Sometimes we use nonlinear correlations, and what you see, like very bright, there was a signal, and the red thing is thing is color models. This is not necessarily related to microscopy, but I think it's very interesting to know and very important. Is a color model is an abstract way to represent colors. And historically and typically, we represent colors by three major colors: red, green, and blue, and everything is just composed of that. Right? It's unfortunately not really lucky because the distances in this color space doesn't really approximate the human eye feeling. Therefore, there are other color models also introduced. CMY, have you ever heard about that? For sure, right? Where? Peters, right? So CMY is called a subtractive color model, where we start from black, white, and we take out the colors, basically. HSV, and there are others which are actually much better or much more suited for image analysis, multiple image analysis, is where we have usually darkness and then a hue and saturation, which goes around with the colors and the saturation out. So in many advanced image analysis methods, these color space is used. So I think that was my first question, what image analysis is, and let's define it. So image analysis is any form of information processing from where the input is an image, and definition of image is a bit, well, it's not very precise here. It can be a set of images, it can be a 3D image, and so on. The important thing is the output is not necessarily an image, but it can be any set of features, and here I put a couple of them. So nuclei detection, deconvolution, and other detection. But license plate number recognition is also something where there is an image and there is an output sequence or medical image processing. Okay? And the most easy image analysis step is histogram transformation. Histogram transformation is a so-called operation. We have a number, we transform it to another number. It's really easy. There are classically from image processing community definition that keeps the original range of values and it's increasing. But basically we have a transfer function which transforms one pixel intensity to another. So let's say we have a picture and this is for histogram transform, which means that darker things are here, brighter things are in pixels are here. And what do you think, what could be the output image if we have this transformation? So dark pixels become black and brighter pixels become the same intensity. So basically what we do, given this transformation, is that we do something like semi-thresholding. Okay? So that's about histogram transformations. Then there are a couple of very interesting and in practice pretty much used histogram transformations. One, which I think the most used is contrast enhancement. So contrast enhancement is nothing more than rotating this transfer function such that we saturate a bit of the intensity range, but we amplify another range. So if Two other histogram transformations which makes the image very good looking, we should be careful with that. One is histogram stretching. Histogram stretching is nothing more, we just pick up the lowest and highest intensities and stretch it as much as we can. So basically we use the entire visible spectrum for visualizing our data. The other is histogram equalization. Careful with that. The idea is that I say that the optimal histogram function would be a uniformly distributed function, which means everywhere exactly the same amount of intensity in my spectrum. This is not very easy to achieve, but it's possible to approximate. So basically what I do is intensities which are not really present so often in my image, I compress a bit, but those which are really overrepresented, I stretch them. And basically what I achieve is like a semi-uniform distributed picture which shows every intensity present on the image with the same ability. Okay. And now a little bit more difficult thing is spatial filtering. So spatial filtering is a way to enhance an image, basically to change the image. And the idea is that we have a kernel, what is a set of numbers you can imagine as a small matrix. We put it to every position of the image and then calculate the underlying dot product and sum of the values. Actually, that was already talking about that for evolution theory. So what I put here is the discrete version of it. And the very nice thing, and actually this is what the microscope does if you have a point spread function and an original function, but it actually happens through the light path. So we can do that artificially as well. And as I said, it's pretty application dependent what you can do with it. But the two major usages are high pass and low pass filtering, and I will also show you a couple of non-linear filters. So let's look what it is. Maybe the first and most easy one is the gradient filter. So gradient filter is a high pass filter, and the idea is that I would like to show the quickest changes in an image. And if you look anywhere and you see an edge, for example an edge here, you see an edge because the intensities which reach your eye are different in this point than this point. It can also be very intuitively generalized for an image. If you would look in this Excel table neighboring pixels which are different from each other, then you would say that there is a quick change on the image. And to express it in a mathematical way, you can create a filter and you can convolve this with the original image and then get the quick intensity changes. So here what we did is we created a filter like that. If you look at it, it's everywhere zero except for the up and down corners where it's minus ones and plus ones. So if you put this to a homogeneous region, it will be completely zero. Whereas if you put it to an edge which has this direction, it will keep a very big response. Or if you put it to another edge, it will give a negative response. Okay? So in this way, we can find certain changes on the image. Let me ask you a question. What would be if we would change this 0 to 1? In a homogeneous regions, there are 3 times minus 1, 3 times plus 1, and we add 1, it basically copies the intensities. Okay? So everywhere where it's homogeneous, we get back the original intensities. Whereas if we are on an edge, we will have the highlights. So if we change this 0 to 1, we got a very nice so-called edge highlighting method, and this is very cool for visualization of changes. Then another one is just take one pixel, take its environment, and average the pixels. What's going to happen is that we get everything blurrish around that pixel. If we use other numbers, then we can get different smooth filters. The one very much used is the so-called Gaussian filtering. The Gaussian filtering, instead of this everywhere one filter, we would use a Gaussian to be Gaussian, and then what happens is that usually the image remains as it was, but quick changes like noise could be suppressed a bit. And then I wanted to show you one more filter, which is called the median filter. Most likely everybody heard about that. The idea is that we just take a certain environment of a pixel and we take its median and copy that. It is actually a fantastic filter because it is capable to remove the so-called salt and pepper noise. So and the salt and pepper noise unfortunately happens many times in microscopy. So the idea is that if we have this very big noise, it's very difficult to create a low-pass filter to filter it out, because an average filter would still keep this noise like quite blurrish, whereas if we apply the manual filter, it really gives us a nice filtering. Unfortunately, big detail or very fine details in the image will reach us down. And the Fourier transform. So I'm not going to give you an introduction or description of the Fourier transform. I rather would like to tell you why is it really great in image analysis. So basically, the Fourier transform has two major usages in image analysis. Very briefly, for those who are not physicists but interested in what Fourier transform does, which we basically say that every periodic function could be described in general with other periodic functions. The combination of other periodic functions, Fourier said that it's the sum of cosine and sine functions. As for example, you see here this rectangular could be very well approximated adding up sine functions with different amplitudes and different phase. Why is it good in image analysis? So normally what you see here is there are smaller and bigger waves. If we have an image, we cannot have small and big waves, but we can have smaller and bigger planes like describing different waves and different directions. And as you can understand, basically an image and its Fourier transform is actually a sum of different waves with different angles, and that if we sum them up, we can get back to the original image. So the representation of Fourier transform is something like that, and it's not important to remember that. The idea is that this is the biggest wavelength, and these are the smallest wavelength, and these are bigger and bigger waves, so more dense waves, which means that this is more detailed information. And the direction around is actually the direction of these waves. So if I would get rid of these very fine things, then I would actually get rid of very dense changes or very quick changes, which is usually either noise or edge. So a Fourier transform can be used to remove noise from the picture and only keep like more fine details or the other way around, just to keep the noise. But that's maybe not the biggest use. The biggest advantage of Fourier transforms is the so-called convolution theory. Again, Gabo was mentioning see image analysis in three major steps. We do some image corrections, then we try to find objects, and then we try to make measurements on them. How much time?
About 10 minutes.
So to get the objects, the process is a so-called image segmentation. And image segmentation, maybe the term is familiar for you, is to find objects on the image, or there are many definitions. I just put what I like the most, is dividing the image into regions that hopefully correspond to the structural units in the scene. Okay, so what we would like to find is structural units in the scene. So for example, if I want to detect bodies, then it would be good if the machine could find me and not this whatever paper. Okay, so the structural units. And here I put a couple of great challenges which are difficult image analysis. For example, medical imaging. This is a so-called energy minimization method where we define the problem as a functional and we try to minimize it. It's a really great idea. Or here, just try to imagine here the letters are more bright. Here the letters are more bright than here the foreground. So and applying a simple thresholding would just kill our analysis. And the big question is how to get a decent segmentation. Okay. The classical approach is thresholding. Thresholding, I think everybody knows, is that everybody who is higher than intensity is foreground, everybody who is lower is background. But the other thing is edge-based. I already shown you like edge highlighting. Edge-based methods are based on edge highlighting, and edge is something which quickly changes intensities. There are very many classical models and newer ones. So thresholding, global thresholding, I don't want to talk much, is just like a very easy thing. Adaptive thresholding is much more tricky, and at the end, any process what we do is based on some sort of threshold. So I should say that this is the most important thing in image analysis, to find a good threshold. However, it sounds very weird, and we've read hundreds of pages of equations. At the end, it's thresholding, and we need to find a good method to find this threshold. So, for example, threshold selection can be something that I would like to try to find two peaks. If you remember when I was showing you the histogram, it usually has two peaks. I would like to find the best separation, which somehow separates them according to the criteria. For example, the isodata criteria, which tries to maximize the entropy between the two groups. Edge detection. So many times I've said an edge is something where we have a quick change. To find a quick change is not very easy because we can't really define what is quick. What we used to do is rather take the first derivative or even the second derivative of the signal. So erasing, if you have a raising curve, the first derivative is a p, the second derivative is a zero crossing. So if I could find a method which is capable to find zero crossing on the second derivative, I can very efficiently highlight the edges. And to do that, we can take a so-called Laplacian operator of the image, which could be approximated very easily by a linear filter. So having a clever linear filter, we can make very effective edge detection. And I wanted to show you other classical methods for edge detection, which are very much used in practice. These are the so-called directional edge detections, and we try to approach the problem such that we try to find x and y directional or assume now we have objects. I would like to get numbers out of the objects. Maybe the easiest way to get numbers out is to measure the area of an object. And we can think about an object as represented by pixels, but we can also represent an object by its contour as like a polygon. So if it's represented by the pixels, the area is not that difficult. We just count how many red dots we have of green or yellow. And if we have an object represented by its contour, then we would basically, the easiest way is to triangularize it and then it's very easy to measure the size of the triangle. For the perimeter, which might be the other very important thing, is we have a bigger trouble if we represent the objects by their pixels. Right? Because if I say that the perimeter is the number of edges, then the number of edges, then I have somehow shorter perimeter. If I say also the number of corners, then it becomes a bit longer. So actually, just based on empirical experiments, we know that if we normalize the 4 connections or 8 connections, which says only the sides or also the corners, then we can get a good approximation of the length of an object. Then there are other ones. For example, a very important one is centroid. So centroid tells you where the object is located, and orientation is how it is located. Okay? And this is not very difficult to calculate based on the so-called shape moments. These are just inherited from the pixels of the object. And here, I think I just come back on. I would like to talk about something which is the so-called invariance. Let's say we have an object under the microscope. We would like to, if we would like to, or let's say several objects, if we would like to find the small ones and count them, does it matter where they are located in the image? Not really, right? Because a small object here is the same small object there. So that means that we don't really care the center, we don't really care its location. So a measurement which tells me what is a small or big object should be so-called translation invariant. So it should be the same measurement whether an object is here or there. And it should also make the translation invariant. Doesn't matter if this object is located like this or like this, it has the same area, right? So then what I need is again rotation invariant measurement. And for example, if I'm looking for ellipsoids, then that I don't care their size, so then I should be size scale independent. So let me ask you, is orientation translation invariant? Yes. Okay. So then